to email him back. All right. So are you recording, Claire, now? Or
I am now. It is now
want to?
recording so that now you can talk about the fancy USRA.
Okay.
This is just an open office hours invitation. I'm not going to turn anybody's cameras off, or, you know, I'll be, yeah, Jenny, if you want to go get your bees. Yes.
I mean,
yeah, it's open office hours. So
Hey, Jenny.
we're here to hang out.
hey, my main question, I just wanted to know if I should come up there to get my bees that you have, Sam. That's my
Oh,
main
yeah. You know, it's you know that we're kind of overwhelmed, and your problem with your bees is that they need some big, in addition to being a lot of them, although interesting, they need some cleaning. So they're still here, but it kind of depends. Like if you need them in a rush, right now we look at it and go like, you know, let's look at something a little more pretty. Sad to say. So what's the status for you?
I gave my final report to the Park Service for what we had already identified. So
Yeah.
I was thinking that one thing I could do, so if that's the issue, I could possibly come up with some folks and we could do some cleaning, if that would be helpful.
Yes, and I've never really done, maybe Mike has some ideas here, or maybe anyone online has some ideas. So the main issue is mold. During COVID, they got stored and then they just molded up. Different, it varied by group, and some of them are like really moldy. All right, there's thousands of specimens. So anyone have any ideas on resurrecting them so that they're a little more,
I've always used a very fine paint, very, very fine paintbrush, but that's just for occasional individuals. I can't imagine doing that for that many.
Yeah, because we actually did process some that had some mold, I think, before we sent them up there, but I didn't realize that they had that extent of mold on them.
Well, it's not something that happened up here. It's maybe, our notion is that maybe they got clean. So I hate to see what they looked like before, but they're they're not our favorite thing. Let me hang on for 1 second. Let me ask Sydney. Is Sydney here? Outside. So I can talk to her in a second. She knows more about where they are. And I can't remember, you had 2 years, and did we go through 1 year and then just didn't do the other year? I know there was a good year or a better year and a worse year.
Well, I don't want to take up everyone's time here with this too, but I just wanted to check in about them. I think that we've processed about maybe 8,000. Actually, we have like 8,000 identified. So I think.
And then, like, so how many are left? Do you remember?
I think it
was a big collection.
1,700.
Okay. All right. And that's both years or just 1 year?
I think that's just the 1 year.
Okay. And then there was another whole year that we didn't start?
I think so, yeah.
Okay. That sounds right.
I might just get those, the 2018 bees, and we'll process them. Maybe I can actually go through this sequence of videos here and just have folks work on those here.
Well, it would speed up our IDs. So you could do that, go through, get an ID that you think is reasonable, and then it's faster for us if, and this would be generally for people who are working with Mike, myself, and Claire, and other folks. If you do IDs before and you're like, so you kind of are confronted like, okay, well, Mike or Sam or Claire or someone else is going to look at our specimen. So why bother? Why bother trying to ID these? Because we're, not going to do it 100%. But the thing is if you do it 90% or even, 87% or whatever, it puts them in groups. That means it's a lot faster for us to spin through the collections, because we don't have, with something, when you're talking like 20,000 specimens, a lot of the actual time is moving things around.
Yeah.
And so there's tricks like some of them you can just put under at the box as a whole under the microscope to check. Yes, they are all bombus and patiens, or like, oh, that one comes out. Let me take a look. You know, it kind of depends on the skill level. Mike, do you have any thoughts on?
It makes all the difference in the world. If they're rough, I just felt rough sorted. You know, if you've got one box and there's 60 species in it, that's just
Right. Makes sense.
But it just cuts the time in half at least, and then you get something out of it because you looked at them, and that's part of the learning thing, you know.
Yeah. Well, we've been able to identify down to family in the past, but we haven't been able to. We haven't taken the time, I guess, to be able to identify them to species. So
even just to genus, or it's as much as someone can do. Like we have our interns and things just morphosort, like sort it by what it looks like, and that really can be super helpful because you do have quite a range of colors and things that you can look at and sizes that you can look at by eye, and then so generally like the process I think of on doing this is like, oh, I've got a huge collection of bees. We tell, our general procedure would be, don't start until everything's done. Okay, everything is labeled, and now you have a whole bunch of groups in a bunch of different boxes. And then at that point, you do a rough sort by eye into bigger groups, maybe into sorting sheets. So you just throw a lot, you take the boxes and put them into sorting sheets, and then, but not with like the notion that you're doing a final destination. And then with the rough sort, then you take each of the rough sorting groups. Again, we use yard signs and put foam on the back of them. And then now I got like, these are all bumblebee looking things. And now I'm going to sit in front of that big sheet and I'm going to like, these things are all the same. These things are all the same. So again, because there's so many different species and it's kind of overwhelming, if you just break it down into those kinds of steps, it's fun and it can go pretty fast, and you can have people who really don't know anything. And then the third step would be for a group like yourself or someone else would be to now start looking and seeing if you can do things with genus or species, but the previous ones are just really helpful.
Okay. Well, maybe we'll come up and get them and work out these steps this summer, since I'm not teaching this summer, so I can come up and get them.
Okay. Well, you could also come up and maybe spend a couple days and do some of this with, right in the lab.
Okay.
And that way, so that we react really well to people coming in, because then we have to pay attention to them. So
okay, I would love to do that actually.
Yeah. And then we can, while we're doing things, we can talk and whatnot.
Yeah, that would be great. Thank you, Sam. I don't want to take up any more because
no, this is a good conversation because it helps us. Mike, do you have any more to add, like what you want to see from people who submit things for you to ID?
Yeah, just the simplest thing. All the green ones go together, all the tiny ones go together. And even though there might be, a lot of the species in there, it just makes such a big difference to me.
Okay.
Saves a lot of time.
Okay.
Thank you.
All right. Good. Thanks, Jenny. I look forward to talking to you and seeing you and your crew.
Yeah. Sounds great.
Beryl asks in the chat, when do you sort by gender, by sex?
Well, I would say that if you're just doing morpho sorting, it kind of depends. So usually there's similarities between the genders. And if you're really good at eyeballing it, you can just see, oh, this is a different thing. And then the males and the females end up into different parts. But, the other part of it is that like Andrina or some of these big ones, the males and the females have separate ID strings. So separating them by gender is useful, but not as useful as the initial morpho sort. Mike, any?
Yep. Oh yeah, completely. Things like Dialectus, Osmia, even Megachile. I mean, it's easy to eyeball the sexes. I just, knowing that you're going to have to use different keys, and so I just separate them as early as I can.
Yeah. When I'm doing IDs, my process is, okay, I've got a big collection. Hopefully it's morpho sorted. And I just start going through it and I just look at by eye everything for the first round. And it's like, okay, got this, got this, got this. And I ID them. And it's a really nice flow. And if I see something like, I need to look this up or I need to verify or whatever, it gets set aside or it's dirty or something. A lot of times it's like, a little messed up, but maybe later, and then come back and do those. And before I do those, I have now a big pile of things that need to be done. I'll go through and separate them by genus. And that process helps me with some of the dirty specimens, because now I've seen a whole pile of good specimens. And a lot of times it's a pattern like, oh, that's this thing that showed up a lot in the good pile.
And I did the same thing. Yep.
Yeah, because when I see people just starting out, it would make sense is, you grab a specimen. It could be a terrible looking specimen, and then you spend 2 hours working through guides and stuff, and that's rough. It's better always. I give people a 5-minute rule. Like, if you haven't figured it out in 5 minutes, put it down when you're starting out and do another one. And you're going to come back. We all come back to these things. And that way you get to look at fresh things and different characters. Characters show up better. And yeah. All right. So we can come back to User Fulvaherta at the end, or if there's a pause, but anyone, maybe we should just see if anyone has any questions or cool things to talk about, or something to show, a picture or any of that.
Tim threw his hand up.
All right.
Hey guys, I collected some bees on Apuntia in southern Georgia. Super excited, assumed they were all Melissodes mitchelli. And when I ran them through the key, it comes down to it's either Melissodes mitchelli or apicatus. And all the features I'm looking at say it's apicatus. The pronotal lobe has definitely got 2 different colored hairs, which is there's a couplet in Discover Life that separates apicatus from mitchelli, and everything kind of points to apicatus. Does anybody have any experience of catching apicatus on Apuntia?
Never heard of it.
Yeah. And so I think of mitchelli as a sand specialist, but so did you go back to LaBerge or Mitchell and look at where it bifurcates in their key?
I did not go through Mitchell or LaBerge.
Yeah. I don't have enough experience with both of them in my head to tell you what any other additional things.
Yeah, Mitchell is worth going through. LaBerge for Melissodes is super hard. I've gone through for and for others, but yeah.
Yeah, I mean, I think the odds are greatly in favor of being mitchelli. I mean, I can't.
That's what I thought too. But I'm just wondering if some of these features that we say are, mitchelli has all white hair on the pronotal lobe, whether that's actually true or not.
I've learned with all Melissodes there's often enough variation in hair color and enough, so you just have to be aware of it in many cases.
Yeah, I hate.
There should be something else besides pronotal lobe color, I would think. Maybe that's all that's in Discover Life, but I would think in at least go to Mitchell and look at where that split is. I mean, there's only a few species in that group that have that pronotal band, the abdominal hairband right on the rim.
Right.
What, 3 species maybe, or is it 4?
I think it's 4.
Yeah. So, I don't know.
The male,
Maybe Gene.
nice-
If Gene is on, he can look it up for us and throw it into the chat or tell
Right,
us what
so
it is.
male has a fairly well-defined yellow clypeus, but the upper edge and down along the sides, it's definitely got a black band. And that's another thing in that one couplet that separates apicatus from mitchelli is, apicatus sometimes can be even all black.
Right. Yeah, no, that's definitely true. I just don't know mitchelli enough to know whether it also has dark sections on the clypeus, because that's unusual too, right? Most Melissodes are all yellow.
Right.
Mike, do you know?
No, I
enough
don't.
about
I mean,
mitchelli?
that's in my experience, that's a rare bee, mitchelli.
Yeah.
Yeah.
It's not-
I've seen it. I've caught it, I think, on the in the sand hills in South Carolina. No
Yeah, I've
records
never
up
got
here.
it in Maryland. I only got it on our trip to Florida. On the way down, we stopped in southern Georgia. Collected someone on the Puncha.
Yeah, I mean, right. I mean, I think of. I mean, you've described apicatus, so it'll be interesting to see. You know, it could be maybe there's a marsh next door and it was like just taking a drink, but you know, I other than that, I don't know what to say. I mean, we just need to look at Mitchell, see what else they say there, but
Okay. All right. Thanks, guys.
yep.
Okay.
Maybe someone else can look that up and throw it into the chat while we're online. I'm incapable of doing that.
I see
Any
Gene's
other?
here. So Eric has his hand up next, and then Madeline has a question from the chat. Go, Eric.
Hey guys, good question, Tim. I'm going to stick to the Melissodes stuff too. Sam and Mike and Claire, I sent you a couple of months ago now about maculatus male with the black hairs on its face. I had questions about that. I've been swamped, haven't got back to it. But my question is, how do you guys combine keys? So Mike has some keys, Discover Life has some, Karen Wright uses a few that she's helped me out with. How are you guys attempting to bring all the Melissodes keys and information together? And the one thing I like about Mike's keys is he has little side notes, or
Right.
you know, these might be found in the Midwest, or they're little different colors and stuff like that. How do you guys go about that if it's capable
So
of discussing that now?
yeah, so when we started out with the Discover Life keys, we basically, and still do, have the whole national collection. So the idea was to look through Mitchell and other keys and get a sense of, well, what are they using? What are the character sets? And then go and directly to the specimens and score them directly for that, because, you know, a dichotomous key, each time it branches, there's species that may have a character that's useful further down that aren't scored for that now. So we wanted to get, you know, the idea with something like Discover Life, where you have a matrix of things, you need to score, or you would like to score every species for every character. And so this is what we're doing with Mike's keys now too, because he has a often comes up with different approaches, and Karen too. And so we've already started working with Karen on taking her spreadsheet and putting it into the wording format for Discover Life. And so so we're trying to combine these things more directly, but the other way to think about any of this is if you have a species and you're like, I'm not 100% sure, and you don't have museum access, is run it through several keys. And even within a key, it's a weight of evidence. Like we were just talking about this Melissodes mitchelli and apicata, or apicatus. And so you would have to, like, okay, it's kind of it coming out ambiguous, or maybe one character points one direction, another character points a different direction. So what else is there? You read the descriptions, you look at pictures, you try other keys, and then you have to kind of Ouija board this. It's not a straightforward thing. And the last thing I'll say is that, so we're taking mics, and some of these are up already. There's a guy named Francis Mullen who might be on here, and he's disassembling Mike's keys backwards. And so he's taking Mike's wording, and then we acknowledge that this is coming from a Mike Arduser key. We put it into the word format of Discover Life, but it's the same information. And most of the time we can incorporate the notes into there. And so that way you have, so the idea with the matrix keys is you have multiple paths. So you could just say, I'm just using Mike's characters. But the nice thing will be, as we go along, we'll try and get more and more of the existing keys embedded into Discover Life. And now you have like many different paths. That formula you would have to go out and look for the literature, and you can choose, you can run them lots of different ways, click things on, click things off, that kind of thing. The idea always being never guess. Like, I like that character, I'm firm about it, click it, not like, well, if I had to guess, it would be that one. That is a mistake.
I think where
So-
I ran into the issue was like with the bimaculatus males there,
Uh-huh.
you know, the majority of them had the white markings on the abdominal segments, and it had the yellow face hairs or whitish face hairs. And then all of a sudden I found one that it was completely black, no abdominal white, and it was a black hairs on the clipius as well. And I
Yeah.
was like, well, okay, that looks totally different than the majority of them. What is this thing? And I could never find a couplet or a key that would actually take on an all black bee with even black hairs on the face. So like a side note or something like that, that would be nice. But I mean, does that happen often that there's regional areas that they look a little different, you know, in Maryland as opposed to Missouri, as opposed to South Dakota where I live?
Yeah. Mike,
There's
you
some-
want to
There's
talk
some
about
truth in
that? Yeah.
Before I forget though, if you've got a male Melissodes and it's got some black hairs on the yellow clypeus, the first thing that comes to my mind is desponsus.
Yeah.
I mean,
Well, and is it Dentiventris would have some or not? I can't remember.
can't remember that either, actually.
Yeah.
So
Right, but
and-
desponsus, and they would have some, I think, on the clipius, the black is, there's black at the upper edge of the clipial suture, so it's not all yellow. Do I have that right, Mike?
yeah,
Or am
and F1 and F2 are different ratios than in
Yeah,
bimaculatus. Yeah.
Mm-hmm.
So, and you probably would have noticed that, Eric. But
Yeah.
yeah, I mean,
I think the abdomen with the all black hairs, like there's no white, it's throwing me off on both of those species that
What
you just
about
mentioned.
T1? Was T1, did it have any white? First of all, did we respond? I mean, did we send back and say what we thought the specimen was? I can't remember.
Yeah, bimaculatus. And Mike, I'm
Okay.
probably just gonna send you some when I send you some more bees,
Yeah,
eventually, if I ever
yeah.
get around to it, but just, I mean, if it is something that's like a subspecies or, you know, it is bimaculatus, but it is all black, then maybe that's a side note that can be put into a key later.
Yeah.
So someone like me doesn't run into it. And yeah, the two species that you just mentioned, I just opened up their pictures on Discover Life, and I don't think, I mean, it's bimaculatus, except it's all black. I mean,
Yeah. We're talking
Yeah.
female. Are we talking females or males?
male.
Well, males would be all black except for maybe T1 on the abdomen.
Well, again, if you look at the photos on Discover Life, there's a male from Florida, and that's why I asked about the regional stuff.
Oh, yeah.
It's a male. It's obviously a male, but it has white hairs on each segment.
That is a described subspecies.
Florida is.
What, Bella, I think.
yeah.
So and I think it might be restricted to southern Florida. And
okay.
often we take pictures of rare things, so it may be distracting to think that that's what a male normally looks like. but most males, I would say, are all black with on the abdomen, except probably T1 or maybe just part of T1. But I could be wrong about that too. Do you know, Mike?
Yeah, I mean, they do vary, but sometimes there's white laterally, on some of the more apical tergites. Not a lot of it, but it's sometimes there. And when I look, when you look at series of males, I mean, they're not all the same, at least in the Midwest. But there's the hair color just varies a little bit.
Yeah.
Yeah, I sent those photos to you, but Mike, I am going to send you actual specimens. Just so you can get
Yeah,
an
that'd be
actual
good.
look at them.
Yeah, that'd be great. So with respect to keys, the way most of mine are constructed now is they're kind of weighted, so that in a given couplet, the lead character is the one that would inspire the most confidence if you can see it, and then so on down the line. So I try to put 2 or 3 characteristics within each couplet, sometimes more. And it's sort of a descending manner, and that helps me. And with all my keys, they're continually changing. I mean, I'm always messing with them because people use them and they ask me questions and they say, well, what does this mean, and et cetera, et cetera. So I've had many people use them, and that's so I am greatly appreciative of that, and so that's why they get a little better.
Right.
Slowly.
Otherwise, it's Mike Arduser's notes to himself.
Exactly.
No, I appreciate what you guys do with the keys and stuff, especially being kind of a rookie here trying to learn them. But yeah, like I said, I got through that key and I was like, this bead does not look like the other one. So yeah.
I'll tell you, interesting. Go ahead, Mike.
I would say, you never know. He may have something. We just, it's possible. So
I still haven't resolved this one. I put it out to Mike and a couple other people, lists and things, but now I'm kind of I think I sent it to Karen, but I'm not sure if I got it back. But I had a female that was all black, did not have the 2 white patches on the rear end, clearly without them, because sometimes a dirty specimen will the white patches disappear. And I found several of these now, and then there's several other subtle differences. So it's like, okay, this is definitely not by maculatus. And then if you key it through anything, it comes up. It doesn't make any sense. But now I'm leaning towards that it's actually an extremely dark version of Brureellus. So anyway. So there's plenty of puzzles out there still,
especially in that genus.
Yeah, there's all those little Botonii fimbriata, and a couple others that I struggle with. But I know I'm in good company.
All right, thanks, guys. Appreciate it.
Thanks, Eric.
Yeah.
It's always really reassuring to know that you guys struggle too.
I mean, we don't.
No, of course not.
We're white males.
Madeline asks, I was curious if there's any updates about the conservation status of Bombus pennsylvanicus. I've noticed a surprising amount of them in Mississippi, but I don't know how they are doing in other areas. Also, Gene is on, but unable to look up things.
Ah, too bad.
So sad.
Oh, he's
Very
in
sad.
his new house.
Yes.
So, Claire, you're Fish and Wildlife, and they're running the show, right? Do you have anything or heard anything?
No, I'm mostly in on the Rusty Patch conversations. Nobody's tapped me for Pennsylvanicus yet. So I don't know where we are in terms of like ESA actually getting moved forward.
Yeah. Mike's, what's the Bombus pennsylvanicus general status, look, feel, just, observationally, Missouri South?
In the greater Midwest, I always see it, but when I first started working on bees in the Midwest in the 80s, it was arguably, at least on native prairies, the most common bumblebee, if not maybe it and grissycolas together. And now, boy, I mean, it's way down the list. but you still see it, but it's nowhere near like what it used to be. And I actually saw a queen yesterday in my backyard, was carrying pollen and that's and nectaring from lilacs. That's pretty early for Pennsylvanicus. I got a good look at it.
Yeah, that's a weird bush too.
It is.
You know, I see honeybees on it, but almost nothing else. Yeah. So I think
Tim, you
the
raised
story
your
is
hand.
Pardon?
Tim raised his hand when we started
okay.
talking about this.
Yeah, I did not have I have a different question. I have nothing on.
Okay.
Okay, well then
Well,
never mind.
I'll just mention that my impression would be that it's not going to be an endangered species level species because it is still like super regular everywhere. We get them here. We're at kind of towards approaching the northern edge of the range, particularly of the range at which they were regular. And we get them I have them at my house and we get them here at the lab all the time. And to the south, that seems to be like they're showing up in all sorts of things, and probably at a reduced rate, as are several other species. When you look at the information, it's heavily weighted towards the north because that's where most people are, and that's where most of the bumblebee people are, and they do most of their activities. And clearly, the species has retreated from the northern edges of its range, where it was marginal to begin with. So I think these pathogens probably are the ones driving it, have reduced its ability to maintain populations. You know, it's just they're sicker. So they're gone from big chunks of the interior of the Northeast now. And my sense is they're stable to maybe slightly increasing, but certainly when you go into the south, they're all over the place. Go to the southwest, you have the Sonoran species, subspecies, and you find it all the time. iNaturalist, I think John Asher said it was the 6th most common bee species on which, you know, it's big, but 6th most common is a pretty big deal.
Yeah, my impression is that it's being added to swaps, like state wildlife action plans, but that's the only other thing I have to contribute to the conversation.
Yeah. Sometimes those are popularity contests.
Yeah, there's other people. Joel is saying that on INET, Bombus is right behind impatiens in the southern states. Madeline responds that it's one of the more common Bombus in their area, pine forests. Eric sent me a chat that it's one of their top 3. So I guess that's what we all kind of have been unanimous about.
Yeah. So we'll see what happens, but I just really don't think it's going to become an endangered species.
Tim, you want to ask your other question?
Love to. So down in Florida, I came across Sphecodes ignatus, gorgeous bee, very red, nice bump in the middle of the vertex. And I saw that Mitchell said it was a subspecies, but Discover Life says it's a separate species from Heraclei. And so I started trying to find literature where it separated those two again, and I haven't seen that. Is it just hold over from this is what everyone thought until Mitchell said that and nobody agrees with him? Or
no, this is my fault.
Mike is the literature.
Yeah, those two are different. They are separate species in places where there are a few places along the coast where they overlap. They're both have been collected together, and sometimes there's there are forms that are a little darker than redder, but the difference is the punctation on the fronds of the two species. So basically, it's not official. It's never been peer reviewed or published that those two are separate species, but just in my experience, it's pretty clear to me that they are. But they both have that knob on the top on the vertex, and that's a very obvious character. So immediately, and they're very similar in other ways.
They're very big too.
Yeah.
What's that? What are they parasitizing? Agapostumum?
As far as I think that's what we generally consider.
They have to be. There's no lazy glossum out there that's big enough to produce this.
Hmm.
So, is a new Sphecodes key coming out at some point?
I keep messing with it. Yeah, I don't know what you have. I mean, I have. I can send you some. I'm not sure what's on Discover Life. I'm not sure if I've kind of lost track of all that, but I do have keys to the groups. So I have a group key. So the Sphecodes genus as a whole is comprised of a number of different, for lack of a better word, I'll just say subgenera that are very distinct and very discrete. And so that's the starting point, at least from my perspective, in understanding that genus. And then from there, it gets a little easier because instead of this one big mess of Sphecodes, you've got discrete groups. So I'd be happy to send you that group key.
Yeah.
It would
I'd
help.
also be interested to know if you're doing any barcoding on that stuff too.
I am not, but people in Canada have, and in fact, they did it quite some time ago. And I don't think there was anything too surprising that came out of it.
Good.
Mhm.
Right.
So it backed up your general notion of
I
what are
hope so,
species
yeah.
and things.
Yeah.
Yeah. I mean, it's still one of the tricky groups. You get into some of that kludgy stuff down there in the, Cressonii, Coronis,
Yeah, those are,
Atlantica,
yeah.
and
You almost need comparative material to really understand those
Yeah.
little ones.
Well, I'm hoping to start up some barcoding stuff in Sam's lab
Oh,
Yeah.
good.
in the near
and
future.
right, that we've inspired Tim because he's a chemical engineer. Come on.
Oh, good.
And Don said he'd be able to help too. So,
And he's retiring, so
all right, guys.
that'll be so awesome. All right.
Thanks, Tim.
So do we have anything else on the docket, Claire?
Nothing else in the chat right now. And I just looked through the keys that I have of yours, Mike, and I don't have anything Sphecodes oriented in our files.
Okay.
So if you did want to send those, I can get them out to everybody. Does anybody else want to jump in with a question? We got like 20 more minutes. Joel, your hand's up.
Curious if anyone has done anything looking at natural land divisions and bee ranges. In Illinois, I noted that the Xylocopa virginica sightings dry up right on the northeastern morainal division and the Rock River Hill Country division. I was just curious if that had been noted anywhere else. It seems kind of odd since that's a generalist and doesn't nest underground, but curious.
Well, yeah, this doesn't pertain to your particular Xylocopa case, but in Missouri, we're doing a ton of that and trying to understand, what are the Ozark bees, what are the Mississippi lowland species, what are the southwestern prairie bees, and there are definite differences, and we've collected, we've been doing this for now 30 years or more, and so we feel pretty confident that these separations of some of these species into the natural divisions of the state are real, and why wouldn't they be? I mean, it pertains to plants and other groups as well. So, but of course, there are species, Halictus ligatus, Agapas ponderescens, that are everywhere, and they don't seem to have any ecological limits or boundaries. They just are very adept. They're just super generalists, and they can deal with anything, seemingly.
Yeah, it'll be interesting to see your Missouri stuff because you have several different biomes kind of colliding there. Whereas in Maryland, where we're doing the same thing, it's a lot cleaner, right? Ocean, east coastal plain, Chesapeake Bay, another coastal plain, Piedmont Mountains Plateau, all in a line. And oh my God, almost everything, although you're right, some are ubiquitous, but almost everything is either has some really clean cut lines or you could say, well, scattered records on the Eastern Shore, but primarily west of the fall line, that kind of stuff. So it really shows up what the drivers are. We can speculate sometimes, but a lot of times it's not super clear. And what we'll see is like it's a coastal plain one, but then if you jump over to like the Hagerstown Valley, in the bottom there, you might pick up another string of them because that's a similar habitat. So
So,
it's, yeah, really
yeah.
interesting.
So Joel, there's been a whole lot of collecting in southern Illinois in the last 10 years by a bunch of different people. And there are a lot of what I'll just call southeastern species that have shown up there. And, Osmia tulibia, Lazy-glossum cruberimum, and others that we had no idea were in southern Illinois. And the same holds for southeastern Missouri. There's this kind of a Mississippi embayment group that come up from the Gulf Coast, basically, and come up the river and the bottomlands along the river, and that's where you find them.
Yeah. Yeah, and it makes sense. Like southern Illinois, unglaciated hill country, really super different than all those till plains and glaciated soils to the north. And is it, you just have to ask like, okay, well, what's the plant group that really creates those boundaries sometimes? And it's not clear. More plant stuff needs to be done, but it's a start. In a couple of these places, we're just getting enough information to be able to start delineating patterns that like the birders have known for years. Which reminds me of Eucera fulvaherta, right? So
yeah.
I don't know much about it, but I've talked to Catherine Paris and then Coleman Little, and it sounds like it's a swamp species. And so you might pick it up in southeastern Missouri at some point.
Yeah, we have.
Oh, you have? Oh,
yeah,
okay.
In a wooded swamp. Yep.
And what's its food source?
Well, it's been collected in this, it's kind of an uncommon bee. It's been collected a lot of different things, and we don't, Coleman thought that there were some styrax grains in the scopa, but I don't know. We don't know yet. I mean, all those other Eucera are generalists for the most part, so I don't know why this one wouldn't be, but, if you live in a swamp.
They're generalists, but they are long corolla generalists, right? So they're on buckeye and penstemon. I mean, that's the leaning is towards things with long tubes.
Yeah.
And they're not dinking around with Asteraceae. Mostly.
Right.
Clovers, Fabiaceae, that kind of stuff. Yeah. Any other, so we're always interested in hearing people's cool things they found. So let's hear it. Let's hear like, oh, I found, just tell us like, oh, we found this really cool new state record or something along those lines. And, bring them out.
This is the Eucera that they're talking about.
I mean, if that isn't
nice.
It's a gorgeous thing, And
Yeah.
of course it's in perfect condition.
Right.
But and then there's no pollen in the scopa, and he didn't wash it or anything, as far as I know. So that's just the way. That's just the way it. But I mean, what's not to like about that creature?
Right. Look at that big
So
honking
pretty.
nose on the Clypeus all coming out there. That's a really good sign. In addition to just being a spring bee, that you might be looking at a Eucera.
And this is one in the field, from 20 feet away. You know what it's going to be. So it's a good one to search for if you're in the southern part of the U.S.
Right. But, despite it would be the thing that everyone would collect, but it's actually very uncommon in collections. So it can't be a common thing, or it's common, but where people don't go, like wooded swamps.
Right. Yeah.
Yeah. Maybe it's.
Yeah, those Arkansas guys are doing some pretty good collecting down there. All kinds of Andrina triquesta showed up.
Oh yeah, I saw that one too.
Yeah, that's like, huh? That was cool. So
yeah,
good stuff.
If you look, there's all kinds of. I mean, in Maryland, theoretically, it was well covered by the people at the National Museum, who would go out all the time on trains, and they were top of the game and have quite a a thing, but we've added many, many more new species to what they've added. And it'll be, once we actually database all their records, it's going to be an interesting comparison and a bit of an insight into like, oh, are bees declining? You know, because we act in a very similar way to them, which is we're trying to hunt down rare things because that's what we like to do. Tim is part of that gang who has found several new crazy species in Maryland by, you know, looking at the literature, paying attention to what's in the field, like there's something different on a something different flower, and then targeting things like sand barrens and different species of plants that are known to have specialists. And if you're interested in conservation of bees, then that's what you do, right? Because the bees that are in soybean fields and in suburbs and you know, the average place are not things we need to worry about compared to Usera fulviherta and Calides ciliatus and stuff like that.
Back on the DNA barcoding, oh wait, hold another chat. Is anybody doing Melissodes barcoding?
I think Karen worked with barcoding, but she's never, I don't think she's published really her work. But I think her her dissertation is out there, but I don't know that I've looked at it. Mike, do you know?
No, I do not know. You know, I haven't heard of anybody.
Well, she's back in the taxonomy game up in University of Washington, so that would be a good one.
Yeah. Hi, this is Lewis. We had sent some bees to Karen, and she got to a point where she didn't believe her own IDs, and then suggested that do a few DNA barcodes. But I don't know who's doing that. So she was looking at things like Limbus and Fumosus and Humilior, some stuff. But she got to a point where she just wasn't sure. I think based on just some ambiguity in the characters, and she didn't necessarily believe the geographic distributions. One or two of them she didn't think should occur up here in South Dakota. So she's the one that suggested submitting them to DNA barcodes, but I don't know who's doing that, and would do a few specimens. Something we'd like to kind of pursue on some of these kind of tough, you know, cryptic morpho species kind of things.
Well, hopefully Tim will be setting that up, and that's the idea, is to I think with Tim and is to mostly be looking at these tough to ID things. So Melissodes is kind of a good example. When I look at the collection at the Natural History Museum, you know, there's a bunch of Wally LaBerge things and other things, and sometimes there's some situations where I'm like, are these right? You know, so we often, like Karen would be in the same boat. She would take material that she thought was identified correctly to be the base of her understanding on species separation things. But if there's multiple species involved, or the person's concept that was doing the ID was different from someone else's, you know, you build in what we've got right now and ambiguity, and because we don't have that DNA barcode, we can't quite say, okay, well, this is variation, this is incorrect, until, you know, you look at it more. And that's, you know, Mike's probably got the same thing because he's right in there, and he'd be good to send those same specimens to, I would think, because he's had a lot of experience looking. Like, Mike, you've looked at Dakota's material and Minnesota material fairly regularly, right?
Yeah, and more southern stuff too, Arkansas. But
Right.
you mentioned Melissodes fumosus, and that was one for a long time that just, I just wasn't sure what it was. But I feel pretty, you know, I've seen enough now from around. I think I have a pretty good idea of what it is. And they do average smaller, you know, but that in itself, of course, isn't too valuable. But Yeah, I'd be happy to
It,
look.
it can be valuable when you have huge numbers,
This is
right? If
true,
you have an
yeah.
actual collection and
Yeah.
you can start laying these out, that's useful and saying, oh, see that one's standing out as being too big or something.
Yeah, yeah.
But
Beryl, do you want to jump in and say what you said in the chat? It's getting set up for DNA barcoding
Sure.
at your lab.
Yeah, hi, can everyone hear me?
Yep.
Yeah, so I'm starting a lab at University of Kentucky this summer, and I've been doing DNA barcoding in Sarah Coker's lab at Princeton, and I have a protocol that works for all of the bees that we've tried so far. We, of course, focus on halictids, but we've done it with other bees as well, and I hope to get that going in my lab. And we primarily rely on the BOLD database for our sort of, reference sequences. So as long as the species is in the BOLD database, it should work. So I'd be open to maybe helping with that, but also the protocol that we use is very similar to some published ones. So there's no really secret sauce. Yeah.
When you're doing that work, so, and that's great because we need more of those kinds of things. And you're in Kentucky, which is sort of a black hole for a lot of distribution information, and is also a tension zone between prairie and western and mountain. But what I've seen in working with people who have the DNA, I don't do it, but is like, okay, well, you grab some of the BOLD data, you grab from the GenBank, and you look at your strings and then try and lay out the specimens too. And a lot of times that, it's the combo. And then you realize, oh, the one from BOLD may not have been identified correctly initially. And that's a big problem because the only vetting is that I said it was, or someone said it was this thing. And if it's Melissodes, grain of salt on all of this, that your material from BOLD is correctly ID'd. Mostly it is, but it's a really, it's a fun puzzle. We all have to do that when we're looking at distributions too. It's like, I don't think I believe that record. Or,
Yeah, so we've noticed often there are several 100% matches, and you can look at the source of who put the sequence in the database, and we trust some more than others. Depending on the species, it's very, very clear that, a number of experts have said it's all the same species and it's 100% identical match, and the next best match is much, much lower, 97%. I don't trust a lot of the BLAST, like NCBI sequences that are there. I trust BOLD more, and you can sort of tell it that you want only high quality sequences included. And there are some exceptions of unpublished sequences where you can't see the source, but usually you can see in BOLD who put it in there. So if I see that Sam Droege put it in there, then I have a little bit more confidence that it's probably a correct ID and better than I can do. So yeah, there are limitations, of course, but it works. The technique works well, and it's, another data point to help.
Right.
Yeah. Here's one there's one of my favorites to throw out. Algochlorella aurata. I think there's two things in there, and I think I was talking with Lawrence, and so I think I have a set of morphological things that separate them. But and I think I shared this with Mike. I don't know what he thinks of it, but I think Lawrence said there were two kind of clades in Algochlorella aurata. Also around the periphery of Algochlorella aurata are a bunch of tricksters, in this gradiola and Persimilis that can sneak in there. But anyway, I think there is, compared to all the old, remember there were these old papers where they would take the propodial characters and divide them into a million different things from the 60s even. And I think some of that is actually real. Anyway, that's one of my favorite ones.
Related to the Algochlorella aurata, last year, someone in my lab collected some nests, and the pupae in some of the nests were Algochloropsis pupae. And we've never seen this before, and we're looking for more now. So if you know of some nesting aggregations of Algochlorella aurata, we are on the hunt. We found a few in Princeton, but it was in Ithaca, New York. And he collected all of the nest contents, and we sequenced them. And then turned out that the pupae were not errata.
All of them or just some of them?
Some of them.
Wow. So like Algochloropsis might be like a sneaky thing.
We don't know. And we don't know if it was, just very random, but there were adult Algochlorella there.
Yeah.
In the same nest. So very interesting. If anyone else sees that, we'd be curious to hear.
Yeah. I don't even know what to think about that.
I mean,
But that's cool.
I guess they could be wrong, but they didn't align to the genome, and then we looked at their cytochrome oxidase sequence, and it matched to Algochloropsis metallica, which now might actually be Viridula. This was a couple years ago.
Yeah,
there's an Algochloropsis.
Mike knows, but I think Viridula is what people would call Fulgida
In the old days.
in the past or the recent past.
Okay.
Right, Mike? And Metallica retained its name.
Right.
And it's the deep blue one. I think of it as deep blue.
Okay. Yeah. So what we had, which we actually sent to Hawaii for the Genome project with the USDA. Right. At that time, when we sent it, we thought it was Metallica, but Zach thinks it's probably, did you say Viridula? Is that how you pronounce it?
Yeah.
That's a more common bee, and particularly up in Ithaca.
Well, it was the pupae that we sent. So it's this maybe parasitic Algochloropsis situation. So yeah, if you hear about that would be fun.
Well, gosh, thinking about their legs, right? If they're parasitic, you would think that they have really reduced scopy and those other kinds of things. And I don't know. I don't think that.
I mean, I think it had to be sort of like an opportunistic rare event, but I guess, not that many nests have been dug up, so maybe it's not as rare.
Maybe it's parasitic on the tunnel building. Like, oh, you do all the work making the tunnel. I'm moving in. I'm going to do a little apartment over here.
Maybe.
Yeah, fun.
That is very cool. We have we've like just a couple minutes left. I wanted to read this comment from Madeline from a little while ago about they've gotten more Tilothrix bombiformis in western Mississippi, the Mississippi Delta, than the eastern side near Alabama. These seem to respond well to cotton. I think that was when we were talking about where to go hunting.
Mm-hmm. Cotton, isn't it? Is it Malvaceae?
Yeah.
And is that so that makes sense? And do people see that regularly?
Up here, Tilothrix is also at what's that plant?
Rose of Sharon?
No, it's a agricultural thing. Okra.
Okra, right.
It used to be a hibiscus. It's now in a different genus. But yeah, your Tilothrix go to that all the time.
Yeah, I mean, it's the right shape for sure.
Yeah, fits perfect. Yeah. And cotton's not all that different. Big flower.
Right, yeah. So much to learn. I mean, another whole aspect is that sometimes you have to be careful because, if you look long enough at one particular plant, everything is going to visit it a little bit. So, the question would be, are these cotton fields swarming with Tilothrix, or is it like, well, there is nothing but cotton right now in the area blooming. So everything might come for a visit. I don't know how much nectar is available in cotton.
Joel just asked a really good question that I want to make sure we capture before I wrap us up. Doesn't Aphanis and Terricola have a parasitic relationship similar with Ogoclorini?
I don't think so. I mean, they share, I think, Mike, or don't they share the same nest parasites? They'll go in what is Bohemicus now or whatever it is.
Used to be Ashtonai.
Yeah, we'll go to both, right? Isn't that right?
They're records.
Yeah, but they're both clearly, I think, I've never heard anything other than that they're both clearly pollen gathering species. The difference between bumblebees that are and are not pollen gathering are super clear because there's architectural features that the nest parasites don't have that the pollen gatherers do, and so it's not. Well, so anyway, does that make sense?
Those two species are largely allopatric too. at least.
Oh, true.
You
Yeah.
know, Turricola, at least historically, has been much more
Right.
northern than us.
Huh. I wonder if that's why some of the Turricula southern edge, that seems to be moving south.
Hmm.
Yeah, because we've gotten records for in Maryland and Western
Wow.
Maryland, and then in the mountains of Virginia that was new, and
Wow.
they seem to be more common in highlands of West Virginia. And that without a lot of previous records, I'm not even sure Turricula was recorded for West Virginia. And Penn State now has Turricula in the area regularly,
Wow.
which and Fernaldi, which is I think also I think does it also do Turricula? What or is that vagans that it's a parasite of? Anyway, it's showing up really like large numbers now
Wow.
too, which is an odd bumblebee distribution thing.
Cool. Madeline wanted to clarify they were catching the Tilothrix in management areas in the Delta, not in the cotton fields themselves.
Oh, so it's yeah. So it's well, and that makes sense. When I think of Tilothrix, I think of the flatlands and marsh edges, and the Delta at one point was like that. So there's probably still sloughs, but not many, because I visited Catherine a little while ago in the middle of the Delta in Mississippi, and it's pretty grim. But there are lots of ditches and things, so I could see that.
Okay, well, this has been fun. Any closing words of wisdom?
Find cool things.
Get out there.
Yeah.
So inspiring.
Nature is waiting.
Thanks, Clare,
right.
for putting it together, and thanks for
Yeah,
everyone
thank you very
for
much.
attending. And
Yeah,
also
this
we
is-
want fewer shy people, because I see people on there that I know come across cool things and they're not saying anything.
Yeah, guys.
Don't make me call you out,
Very intimidating over here.
Anthony and
Okay.
Mark.
And what are we thinking about next week? Oh yeah, Mark
Well,
is here.
we-
Hey, Mark.
We kind of finished up Megachilidie. Mike, any notions?
Did you ever-
Caletidae?
Did you ever do Steelus?
And we did. You were gone
Yeah,
though,
okay,
sadly.
okay. Glad it was.
Yeah.
Yeah, Caletidae. Yeah, I mean that.
How about Caletidae?
There's a lot of.
We've done
Yeah.
we've
Pardon?
done Calides and Hylaeus
Oh, we did.
already.
Okay.
But
What
there's
about
some other
I
stuff.
can't forget how long we've been doing this.
How
I
about
know. This is like our 80th episode, I think. Something like that.
Yeah.
Well,
Okay.
send us your votes. Email me what you
Right.
want us to work on next. And that'll inform our next decision. Adrena.
We do have Lazy Glossum hanging out there. We should probably bring in some of Jason's people to help work us through that. But
Rob said he would come
It's
for
a-
Andrina if I
And
reached out,
Andrina
but I
too.
don't know how many months he has.
yeah. Well, I mean, we've got some of those big ones. So
Yeah.
I
I'll
mean,
ask him. I'll send him
Andrina
an email
versus
today.
Lazy Glossum. Yeah. And we can split Lazy Glossum up some.
Okay. Well, people have spoken. We got multiple Andrinas on the chat.
Oh,
All right.
All right.
Yeah.
So I'm
All
going to
right.
wrap up the
Fine.
recording and see you all next week.
All right. Thanks, Claire.
Thanks, Claire.
Thank you both.
Thank you, Mike.
All right, Sam, get out there, collect.
All right.